this is Miss Dagenford again, and I wanted to tell you a little bit about the structure of DNA. The other week, we were looking at the experiments that showed that DNA was the genetic molecule. And so, through all of those experiments and research, we finally realized that yes, DNA is the genetic molecule, but then was the race to find the structure. And we finally realized through the work of Rosalind Franklin and Erwin Chargaff and Watson and Crick and others that we did not go over, the shape of the DNA. So that's what we're going to go over today. So let me go ahead and present this. Now make sure as you are going over your Google Slides that you update your slides so you get the same information. Now, DNA is made of nucleotides, and the nucleotides are going to be connected to each other to make one long polymer. Now, we talked about polymers back in September when we talked about proteins and polysaccharides, and then we talked about DNA, and DNA is a polymer. It's a polymer of nucleotides. All nucleotides, sorry, bad typo there. All nucleotides have three parts. There is a deoxyribose. It's a 5-carbon sugar. And then there's a phosphate. And these are the ones that end up connecting to another nucleotide. And then the base, the nitrogenous base, is then going to be on the inner part of the helix. And that is a nitrogen-containing part of it. Now, there are two types of nucleotides. You can see on this one, there are two rings, and we call those purines, adenine and guanine. And then we also have pyrimidines, and pyrimidines are only one ring. Notice thymine only has one ring, and so does cytosine. And so what ends up happening is you've got one nucleotide, here's thymine, connected to another nucleotide, here's guanine. That makes up one side of the DNA molecule, and then it is connected to the other side through the pairing of the bases. The thymine will always match to an adenine, and then a guanine will always match to a cytosine. Now, the strand is then twisted in the form of a helix. There's about 10 bases per turn of the helix. The sides of the ladder are made of the alternating phosphate sugar backbone. So phosphate deoxyribose. So here we have one side. Notice my cursor here. You have the phosphate and then the sugar of one nucleotide connecting to the phosphate and sugar of the next nucleotide connecting to the phosphate sugar of the next nucleotide. So the sides of the bases are phosphate sugar, phosphate sugar, phosphate deoxyribose, phosphate deoxyribose. Now the two strands run in the opposite direction, and the way you can tell that on this diagram is notice the point of the sugar would be facing downward here. So this strand is pointing up on the left-hand side. The other strand is kind of running downward. Now it's going to look similar. The strands, the backbone is still going to be deoxyribose phosphate, deoxyribose phosphate. And then the bases are in the middle. Now you are going to then have this run for thousands upon thousands of nucleotides all the way down. So the sides of the ladder are going to be connected to each other by strong covalent bonds. The inside rungs are going to be hydrogen bonds. Now, a quick review of those would be our partial positive and partial negative, positive and negative charges. And I have another picture of this on the next slide. So here would be our thymine, and notice our thymine is attracted to the hydrogen and nitrogen of the adenine. And so these dots in between them represent hydrogen bonds. Now, those are just attractions to each other. So collectively, think about a Velcro, piece of Velcro. One piece of the Velcro you could tear apart pretty easily, but if you had all of those hooks together, it's going to be tougher to rip them apart. So one adenine and thymine to the other, hydrogen bonds are weak, but if you have a whole DNA strand, they're enough to keep it together as a double helix. Uh, so we have these base pairing rules between adenine and thymine. Every time you see a thymine, it's going to be facing an adenine on the other strand. These are complementary, not complimentary like you're giving somebody a compliment, but complementary like you're completing it. So just like a puzzle piece is complementary to another piece of the puzzle, adenine and thymine can only bond to each other nicely. They've got these lovely two hydrogen bonds that are going to fit together nicely. And then we have guanine and cytosine, which is going to pair together. And you can see they make three hydrogen bonds. They're a little bit stronger for that reason. We have three hydrogen bonds occurring. And so remember, that means we have a partial negative charge on the oxygen, partial positive charge, and that means they're attracted to each other and they're holding their place. So what do we notice here? We've got our phosphate on one nucleotide, a deoxyribose, and then our thymine base connected to, on the same side, the phosphate and deoxyribose of the guanine. Now, this would continue up and down. They're just showing you a little picture. Then we have the bases in the middle, thymine binding to adenine through those hydrogen bonds, guanine binding to cytosine through those hydrogen bonds. And then on the other side, we've got our base, our deoxyribose, and our phosphate connecting to the deoxyribose and phosphate of cytosine. So there's your basics of the structure of DNA. Now, you're going to see, we're going to do some practice with these movable pictures. And so you'll notice that these are kind of put together to kind of represent the molecular structure. Now, they can be a little tough to visualize, right? Here's what they actually look like. The orange is going to be phosphate. So we've got our phosphate, our deoxyribose, and then we've got our base. And then we've got our phosphate, and then we've got our deoxyribose, and then we've got our base. And notice all of the bases in our little cartoon picture have what look like a piece of a puzzle connected to it. So notice here's our G, there's a little puzzle piece. Our A, here's our puzzle piece. And then our C. And then we've got the arrows. And just like pieces of the puzzle, the arrows are always going to go in the empty arrow spots. So what I want you to do, and you guys can do this together as a class. Remember our base pairing rules. I want you to go through this strand. Here is one strand of DNA. And we're just going to practice our base pairing rules. And I've got a whole bunch up here. They're all stacked. And so I want you guys to, and what you can do is pause the video, decide as a class what should go there. And you'll notice that, again, just like a piece of a puzzle, if you tried to think, oh, well, here's a T, well, notice the T doesn't fit. It doesn't match. So we can put that one back, and let's take a look at G. Well, even if you wanted to make it work, G and T don't fit like a piece of a puzzle. So let's put that one back. So hit pause and see if you guys can decide what is going to work. And so hopefully you decided that A is going to work, and it's going to take a little bit of practice to get these perfect. So let's see if you can get the next three. The next three are C, T, G. Decide, pause now. All right, let's see. Hopefully you caught the fact that it was G. And then we have another A. And then we should have a C. And it's going to take a little bit of practice to get these perfectly aligned. All right, you know the drill. Let's try the next three. It's A, C, A. Go ahead and hit pause. And hopefully you picked T and G. And then we should have another T. Let's get another yellow one. And you can, of course, if you need to, you can fix these guys. Should be nice and level, but they're not quite as easy to do with the jam. They're easier in the Jamboard to kind of manipulate. There we go. And then finally, let's do the last one. We have T, G, and C. Hit pause. And hopefully you are getting it, getting the idea. We have A, and then C, and of course you can see if you don't get it perfectly right, if they don't match up, but they're not too bad. And then another G. Let's get that up. So try your best to get these. The idea is you want to be able to notice that they're puzzle pieces that fit together, so it does work better the closer you are to matching it. So don't skimp on that. Try to get as there we go, as good as possible with our bases. And of course, I'm having a hard time there. But notice our base pairing rules then gives us a way to know what the other side will always look like. And that's the pivotal part of our DNA replication, which we're going to do next. So just a reminder, you should have all of those slides done for all of our experiments, and we just did slide 57. Now, as a class, go ahead and hit pause, and you're going to do this section together, and then we'll go over it in the next video.